Hello everyone, welcome to today's webinar. I'm Kübra from IBM Technology and I'm delighted that you could join us. In this session, we will be sharing our latest optimized intravital imaging protocols developed and refined with direct input from researchers like you. And these protocols address common challenges in intravital microscopy, such as complex surgical preparation, inter-user variability, and the step learning curve that often limits broader adoption. Our presenter today is Hayden Kwon, as some of you already know her, and she is a senior application specialist at IBM with extensive hands-on experience in the intravital imaging. As you listen to her presentation, feel free to visit our website to explore the imaging systems and the accessories we will be discussing. I will also share the link in the chat box during the webinar. And please also feel free to submit your questions at any time through the Q&A box below. We will make sure to address as many as we can at the end of the session, and also to those we received prior to this webinar. Hayden, if you are ready, please begin with your presentation.
Okay, so good morning and good afternoon, everyone, and thank you for joining today's webinar. So today, like you said, I would like to introduce IBM Technologies' Intravital Microscopy platform and walk you through several of the standard and accessible surgical techniques that enable its successful application. For the start, please let me briefly describe the fundamentals of intravital microscopy and highlight several key features that are unique to IBM's imaging platform. Intravital microscopy is a high-resolution imaging technique that enables us to observe subtle biological changes inside a living animal down to the level of cells and tissues in real time. By comparison, traditional histology methods rely on fixed tissue sections. So while they provide detailed structural information itself, they only capture static uh, snapshots, making it impossible to track dynamic processes as they unfold. Similarly, in vitro techniques using cultured cells allows for controlled experiments but fail to fully replicate the complexity of the in vivo environment where cells constantly interact within their native tissue and vascular networks. Other in vivo imaging modalities such as PET, CT, or MRI are valuable for analyzing physiological changes at the tissue or organ level, but also they lack the resolution needed to visualize cellular level events. In contrast, intravital microscopy fills this gap. So it allows researchers to continuously monitor drug distribution, cellular interactions, immune responses, and disease progression directly within a living subject. So this unique combination of cellular resolution and real-time tracking provides the more accurate and physiologically relevant understanding of biological processes. Like I mentioned before, traditional techniques only yield rough, raw resolution data since they capture images at the whole body or whole organ level. So, to address these limitations, we have developed a more advanced intravital microscope that enables researchers to track various cellular dynamics, monitor drug distributions, and assess therapeutic effects even in challenging organs such as lung and heart, which I like to show you later in living animals. So, a defining strength of intravital imaging system itself lies in its clear departure from conventional in vivo approaches. Traditional methods typically require sacrificing separate animals at each time point, which inevitably introduces substantial inter-animal variability. In contrast, intravital imaging system, especially ours, allow longitudinal imaging within a single living subject at precisely defined intervals, dramatically improving consistency and reliability. So beyond static image acquisition, it enables real-time dynamic visualization of biological processes as they unfold, offering a far richer and more accurate perspective on complex physiology. To really understand how our body works, we need to see what's happening inside, right down to the level of the cells. So this is especially true when we study the immune system, cancer, or how medicines move and act in the living bodies. So intravital microscopy makes this possible. So it lets us watch these changes unfold in real time inside the living animal. So our system in particular allows us to track how drugs travel through the body and how they affect cells life as it happens. For example, on the left, you can see the data from a sepsis model where inflammation blocks blood vessels in the lungs. And on the right, we show how our imaging system track exosomes, anti-cancer drug distribution, and transplanted cells moving through the body. So IVIM's intravital microscope are designed as standalone systems, making them easier for users to operate. So these systems are simple to insert and maintain, allowing researchers to focus entirely on imaging and experiments without limitations from space or other environment conditions. So it still delivers outstanding intravital imaging performance through our in-house developed hardware and software. So these include the ultra-fast polygon scanner, a real-time temperature monitoring system, motion compensation, and AI-based denoising software enabling high-resolution imaging of diverse organs in living animals. So among these features, the motion compensation system corrects for organ movement so researchers can obtain sharp and accurate images even in high dynamic organs. The real-time temperature monitoring system ensures that an animal's body temperature is continually tracked and adjusted if needed throughout long imaging sessions. So this not only minimizes physiological variability caused by temperature changes, but also maintains an ethical sound experimental environment. Finally, our released AI-based software preserves true signals while effectively removing only the noise. So this provides cleaner, more reliable data that can be used directly for quantitative analysis. So later in this talk, I will also show you the example data sets. Okay, so one of our powerful key features, the motion compensation, I like to address it more. So this is in vivo real-time video of kidney. So in this side-by-side comparison, raw imaging data has high degree of movement. So without motion compensation, only this blurred image can be obtained due to motion artifacts, which highly disturbs the quantitative cellular analysis of the data. But when we apply the motion compensation function, we can directly acquire the high quality of clear image without any shifts. Thereby, IVIM has optimized the in vivo solution for intravital heart imaging or lung imaging in cardiac and respiratory research fields. Recently, through joint research, an AI-based noise canceling function has been developed and is and released. So even if the motion compensation you forgot to adjust the motion compensation algorithm, this function preserves frames at the same raw video speed even in real-time video at 30 to 50 frames per second. So this is an example applied to muscle. So the actin and myosin structure is clearly seen, and RBC red blood cells flow at a very fast speed also could be visualized in high resolution. And for sure, after acquiring the heart imaging data, we could improve the quality of the single image a well of movie data. And I like to emphasize that we have fastened this speed of the denoising procedure with over 20 or 50 times of the training. And also, we have only licensed to utilize this kind of motion the AI denoising software algorithm in the market. So one of another key features of our imaging technique is the windows and chambers, which are called. So IFIM's intravital microscopes are designed not only for short-term studies, but also for long-term imaging applications. So to make extended imaging more stable and reliable, we provide a variety of window and chamber options. So by using these chambers, it is possible to perform PK or PD analysis, tracking disease progression or drug movement in real time within the same living subject. On this slide, you can see some of our most representative windows and chambers. So if you have a particular research interest in a specific organ, we can also develop and optimize the customized ones for you, tailored to your study needs. So if you have any inquiries, please contact us. And with a microscopy platform specifically designed to capture dynamic cellular behaviors in living animals, alongside a range of specialized imaging windows and chambers, you can obtain high-quality data sets like the ones shown here. So these tools enable the visualization of diverse immune responses, disease onset and progression, as well as the drug distribution and therapeutic efficacy, supporting a wide spectrum of biological and pharmacological research applications. So from here, I'd like to guide you through each application step by step, beginning with the brain, then moving on to how we achieve live imaging in highly dynamic organs such as the lung and heart. Finally, I will also show how these imaging windows could be applied not only in mice, but also adapted for use in rats. In intravital brain imaging fields, researchers generally have two major needs. The first is imaging of superficial regions such as the meninges or cortex, and the second is assessing deeper structures, including areas like the hippocampus. So I will explain those two approaches in sequence. So first, uh, the description of cranial imaging window implantation surgery to visualize the surface region of the brain. So to give you a clear overview of the procedure, I have prepared a short video demonstration here. So after removing the hair with clippers and depilatory cream, the mouse is positioned on the surgical stage. And we then incise the scalp and thin or remove the skull over a 3 mm diameter area with a hand drill piece. And after microbleeding stops coming from the bone itself, a 3 mm diameter cover slip is then placed over the exposed cortex and we secure it using various adhesives. Loctite and other bonds. Finally, the dental cement. So this Windows maintains over 1 year and a half. So this tool is very useful for studying many brain-related diseases such as Alzheimer's or Parkinson's disease and brain tumor as well. So shown here is the overhead view of the completed cranial imaging window. Now here is one of the brain imaging data sets, and I'd like to walk you through how our system can be applied in real experimental settings employing the cranial window implanted mouse models. Uh, this will give you a clearer sense of the system's capabilities and how it can support your own research. So in this study, we induced photothrombosis by administering rose-spangle and subsequently irradiating the tissue with a 561 nanometer laser. So by staining with DHE and PI dye, we were able to visualize reactive oxygen species and cell death, confirming that the photothrombotic injury was successfully established. And further, we visualize the same location of the brain to see the cerebral vasculature changes from day 0 through day 30. So following a photothrombosis induction, we initially observed signs of brain swelling, likely driven by acute inflammatory responses. However, as we continued longitudinal imaging up to day 8, a notable shift became apparent. The brain exhibited even greater contraction compared to day 0. So at this point, we wanted to understand what cellular mechanisms were occurring during the period when brain edema rapidly resolved, specifically between day 2 and day 5. To explore this, we conducted the following experiments, which was the evaluation of vascular permeability by injecting fluorescent dyes of different molecular sizes. The smaller 3 kilo Dalton Tri-TC dextran dye across the blood-brain barrier much more rapidly than a larger 2 megadalton FITC dextran dye. So this differential leakage pattern indicates that the BBB was compromised following photothrombosis and microinfarction, resulting in transient vascular disruption. Interestingly, by day 5, the leakage of the tri-TC dye had returned to baseline levels, suggesting a gradual recovery of blood-brain barrier integrity over time. So at this stage, having observed signs of BBB recovery, we aim to identify which cell types might be contributing to this restoration. So to do so, we used mice expressing fluorescent signals, specifically in NG2 positive parasites and in the nuclei. And also, we administered an in vivo vascular labeling kit to visualize the vasculature, and you can see them in blue color here. And we then performed repeated imaging of the exact same region over a 22-hour period. So as shown in the video on the right, we were able to directly observe NG2 positive cells, namely parasites, elongating and dividing along the cerebral vasculature in real time. So this example highlights an essential strength of intravital microscopy. It does far more than simply allow us to observe cells. It enables us to watch how those cells behave and respond in real time. As a result, we can continuously track disease onset, progression, and restoration as they unfold within a living organism. Beyond observing cellular responses within the mouse brain, researchers can also establish fluorescently labeled brain tumor models by injecting tagged cancer cells into the brain. So this enables the investigation of a wide range of pathological and pharmacological characteristics related to the brain tumor development or the treatment. So in this model, we injected glioma cells before securing the glass cover slip and completing the window implantation procedure. So 7 days later surgery, we confirmed that the tumor cells had successfully engrafted and begun to grow within the brain. So following tumor establishment, we administered a nanoparticle-based immunotherapy drug intravenously. So you can see the drug in red color here. And this allowed us to directly evaluate the therapeutic response by observing immune cells labeled in white in these pictures, successfully infiltrating into the tumor-bearing brain tissue. So through this dataset, we were able to obtain clear, real-time evidence of the drug's efficacy. In addition to these applications, we also sought to evaluate the real-time effects of analgesic compounds by monitoring stimulus-evoked neuronal activity. So for this experiment, we implanted a cranial window in Thailand GCamp 6F mouse to visualize calcium channel dependent neuronal responses. We then applied bone-free filament stimuli to the hind paw to induce sensation and pain while performing live imaging of cortical activity. So following the stimulation, we observed a clear stimulation and the administration of the drug, we observed a clearer decrease in the frequency of activation signals after the drug distribution administration. And this demonstrates that intravital microscopy allows us not only to visualize cellular movements or division themselves, but also to directly monitor functional activation patterns in real time. In other words, we can capture how cells and even neural circuits respond dynamically to stimuli and treatments, providing a far deeper and more comprehensive understanding of biological processes in vivo. Traditionally, imaging the hippocampus required ex vivo approaches, either uh, clearing the extracted brain so that the laser could physically reach the hippocampal region, or sectioning the tissue to expose the area of the interest. However, both methods inherently compromise the physiological environment. So to address these limitations, we optimize the surgical procedure and an intravital imaging workflow that allow direct visualization of the hippocampus in vivo. So the surgical approach begins very similarly to the standard cranial window surgery. So after removing the scalp hair and securing the mouse on the surgical stage, we incise the scalp and drill the opening in the skull. So next, the cortex overlying the hippocampus is carefully aspirated to physically expose the target region, and a cylindrical cannula with a glass cover slip attached at its tip is then inserted into the cavity in this way, and then cannula is secured in place using adhesive, completing the implantation procedure. Once the mouse is mounted on the imaging system, and the scanning begins. We could visualize the hippocampus. So in this study, we aim to examine changes in immune cell distribution, particularly neutrophils within the hippocampus, by inducing a drug using an approach similar to the previous dataset. So after labeling the blood vasculature and neutrophils with in vivo labeling kits, we performed imaging before drug induction and continued up to 24 hours afterward. So by looking at the vascular structure, you can see that we consistently imaged the exact same region over time, and the vessels became noticeably atrophic, confirming successful drug induction. And we also observed a substantial recruitment of neutrophils into the affected hippocampal tissue. So because our IBM system provides image data of sufficiently high quality for both qualitative and quantitative evaluation, it also enables precise numerical analysis as shown here. And this concludes the overview of surgical techniques for imaging various regions of the brain. So next, I'd like to introduce our approach to intravital imaging of the lung and heart. So traditionally, imaging these highly dynamic organs has required sacrificing the animal, as their constant motion makes live imaging extremely challenging. However, we have addressed this limitation using a simple negative pressure stabilization methods. So lung and heart imaging begins with the endotracheal intubation, which is performed immediately before surgically opening the thoracic cavity. So after making a small incision on the ribs, we can observe directly the lung or the heart expanding with each breath or beating. So at this stage, we position the lung or heart imaging chamber connected to the gentle vacuum source to stabilize the tissue and minimize motion to acquire high resolution imaging from both of the organs. The lung and heart definitely are of the most challenging organs for lifestyle imaging, largely because they are constant motion due to the respiration and breathing. Sorry. So to address this, we use the TMS, which I called the simple and the gentle vacuum-based source. to gently hold the lung and heart in place. So by applying controlled negative pressure, the device effectively immobilizes the tissue, enabling the acquisition of high quality, stable images. So with this setup, we were able to capture real-time of cells moving through pulmonary vasculature as shown on the right video, and we could also perform long-term imaging without losing stability. So in this application, we aim to visualize dead space formation and reactive oxygen species release in a sepsis-induced lung injury model. So within the pulmonary vessels, you can see rapidly moving erythrocytes labeled with DID as they circulate through the microvasculature. And on the left, red-colored neutrophils are shown accumulating in the branches region where an arteriole transitions into a capillary. So these cells form a distinct cluster reflecting the inflammatory response characteristic of sepsis-induced vascular injury. So which is called the dead space. So these cells form a distinct cluster reflecting the inflammatory responses, and as a consequence of this clustering, that space was generated within the affected region of the pulmonary microvasculature. However, we also observed that these dead spaces disappeared once microcirculation improved following drug treatment. So, in other words, even in organs with substantial motion, it is possible to obtain stable long-term visualization data and, importantly, to clearly capture the immediate therapeutic effects of administered drugs in real time. Next, to show you a more dynamic example, I'd like to present imaging data captured from a coronary vasculature, highlighting both cellular movement and lipid accumulation observed within the heart. So with our high-speed scanner operating at 50 frames per second, we can clearly resolve individual cells rapidly flowing through the bloodstream without any motion blur as you saw earlier. So this not only enables precise visualization of fast-moving cells, but also allows accurate measurement of blood flow velocity. And the heart imaging chamber, which is highly similar to the lung imaging chamber, not only stabilizes the beating heart for observing rapidly moving cells, but also enables researchers to acquire high-resolution motion-free images. So by using a specialized labeling kit designed to stain lipid droplets in vivo, we were able to visualize lipid accumulation in the heart following a high-fat diet as shown on the right. And as you can see, prolonged exposure to the high-fat diet leads to a gradual increase in lipid droplet accumulation, and by the later time points, we also observed a higher number of infiltrating immune cells compared to 4-week stage. So this suggests that progressive lipid deposition can trigger a range of cellular responses within the cardiac microenvironment. So in this way, IVIM's intravital microscopy platform enables far more than simple cell observation. It allows researchers to visualize a wide range of biological responses that accompany disease progression or therapeutic intervention, providing a deeper and more dynamic understanding of in vivo physiology. Lastly, I'd like to describe the similar internal organ visualization with specialized windows optimized for rats to tailor to the needs of utilizing these advantages of intravital imaging techniques from rats. The procedure is carried out in the quite same way as in mice. So this rat was anesthetized with ketamine, or also you could utilize the isoflurane as well. So once anesthetized, the abdominal hair was completely removed using clippers and creams, and then the skin then incised to fit the size of the window. Next, the window was secured to the skin and the peritoneum using a purse-string suture technique, and the surgery was completed by attaching a rounded glass cover with adhesive. After completing the abdominal imaging window implantation, The rat was placed on the free space system's mounting stage, which is especially designed to perform the imaging with rats or ferrets. And the window also securely was fixed in the holder. And the FITC dextran dye was then injected via the tail vein route to visualize the blood flow. To maintain the physiological conditions, a heating cable was connected to the system for temperature control and monitoring. The stage position was first roughly adjusted with the handles and then fine-tuned with the jog dial to locate the focal plane before proceeding the imaging. And this liver observation through the abdominal imaging window in planet rats, rightly same with the procedure on mice, also provided stable imaging conditions. So this allowed us not only to capture the deep images, but also to acquire a deep and wide field images using mosaic imaging. So with video level imaging, we could clearly visualize the blood flow, which also the possibility of measuring blood flow velocity. So now you are looking at the data before and after processing with an AI denoiser software and with the middle-sized animal imaging platform together with the AI denoiser. I'd like to introduce these powerful tools that enable the observation and analysis of diverse biological phenomena in rats as well as in mice. So currently, we are expanding the pipeline to include targeted cell lifelink and other organ imaging to monitor cellular responses in various disease models, and this will allow us to share an even wider range of data set in the future. Okay. To conclude today's presentation of mine, I'd like to briefly summarize the key points we discussed. IVIV's intravital microscopy platform offers far more than single time point imaging. So through a variety of specialized windows, chambers, and organ-specific approaches, including those designed for highly dynamic organs such as the lung and heart, which I described in previous slides, we enable researchers to access regions that were previously difficult or even impossible to visualize in vivo. And at the same time, integrated systems that maintain body temperature and support optimal physiological conditions ensure that the data obtained are truly representative of in vivo biology, allowing for high quality, reliable imaging across a wide range of applications. Also, from basic scale systems designed for small animals like mice or hamsters to larger series that can even be applied to non-human primates, our portfolio covers a wide spectrum without size limitations. Through this diverse set of intravital imaging platforms, I hope you could appreciate that we are not merely observing cells, but gaining the ability to visualize the organic, dynamic, and truly living behaviors of cells within their native environment. And thank you for your attention, and please leave your questions in Q&A box. Thank you.
Thank you for the excellent presentation, Hayden. The first question we had previously is that are there any training programs or resources available for new researchers to quickly learn these protocols?
Absolutely. So we offer both online or and on-site training programs upon your request, and all sessions include the hands-on practice as well. So the typical training session lasts about 2 to 3 hours, I think, and most trainees are or able to acquire the publication quality images within 3 to 4 practice sessions. And also, it seems like very difficult to perform, but like I said, I like to emphasize that just 2, 3, or 4 practices would be enough for you to get used to every surgical steps.
Thank you for the answer. And another question is about more um, the equipment and it's a technical one. So what is the temporal resolution and frame rate achievable with your multicolor real-time imaging setup during in vivo experiments?
Mm. Okay, so
Temporary
our
response rate.
mm, okay. So our system has the high speed ultra fast polygon scanner, so the frame rate will be 50 frame per second up to, and it can capture four uh, independent separated fluorescent channels simultaneously. For example, DAPI, GFP, RFP, and Sci-Fi with a confocal laser. But one of our most widely famous models, the CMS3, is equipped with both a confocal and 920 nanometer fixed wavelength two-photon laser. So even with this fixed two-photon wavelength laser, using a single wavelength 920, you can still capture all four channels at the same time.
Mhm. And another question is, how do you handle photo bleaching or phototoxicity during prolonged imaging sessions, especially when tracking multiple fluorescent channels?
Okay, so let me say that our system, firstly, is designed for the in vivo imaging itself. So the laser power itself is intentionally kept at very relatively low levels, minimizing the bleaching and toxicity effects. So that's said to further reduce any potential adverse effect. We we and suggest some kind of specific imaging conditions, including lowering the laser power as needed based on the user's experimental requirements, and which is called the sequential imaging mode. So you can capture all four different colors simultaneously, but very, very close to the sequentially. So which means you could reduce the effect of phototoxicity or other bleaching effects.
Okay, and lastly, I think this is the last question. Do you have any strategies or software tools do you recommend for real-time quantification of platelet aggregation, immune cell trafficking, or vascular remodeling?
Okay, so in terms of the platelet aggregation, immune cell trafficking, or vascular remodeling imaging, maybe you need to acquire the real-time imaging data, I think, and also you have very huge data sets. So for the imaging analysis, we typically recommend ImageJ for basic quantitative or qualitative analysis, and IMARIS or ARIVIS for more advanced complex data sets. So all images acquired with our system would be saved in formats that can uh, be compatible with those two or three uh, softwares. So there is no need to convert the imaging formats to utilize that kind of softwares. But also, we have our own in-house software, which is called Studio for the post-processing the acquired images. And also, very early next year, maybe we could add some functions for like a basic uh, analysis for measuring the length, to see the dilation of the vasculature or count the platelet aggregation, or also you could simply measure the velocity of the immune cell movement in the living body.
Okay. I think we answered all the questions for today. If you have any more questions, please feel free to ask us through email, information@ibmtech.com. And thank you all for your questions and active participation. As mentioned earlier, you can find our imaging accessories, fluorescent labeling agents, and training programs on the IBM's website. And if you create an account in the shop page, you will also gain access to member exclusive benefits for the future purchases. And for any additional inquiries, please feel free to contact us at information@ivmtech.com. And thank you again for joining us today, and we look forward to seeing you at our next webinar.